Good afternoon, everyone. Thank you for joining. My name is Wenzhi Tian. I'm the founder, chairman, and CEO, and also CSO of this company, ImmuneOnco Biopharmaceuticals. Our company went to public in September of 2023 in Hong Kong market. Please forgive me if I cough a little bit because I got cold. Yeah, this is our company's development journey. So we incorporated in June of 2015, and 8 years later, we just went public. So this year is 10 years anniversary. And so today I'm going to present several important assets. I'm sorry. So since we are an incorporated company, so we have integrated the proprietary R&D platform, including target selection, validation, and drug discovery, and also high-throughput screening. And also, importantly, we have our own pilot manufacturer, including 200-liter and 250-liter bioreactors. So this is our pipeline. So covering two aspects. For the first part, covered by a light blue is oncology area, and below is for a non-oncology area. So I'm sorry. No point. So if you look at the two assets, program names IMM2510 and also IMM27M. Those two assets has been already licensed to InstillBio last year. So I'm not going to talk about these two assets today. The first one, which is IMM01, is a CD47 targeted molecule. Right now, we are conducting phase 3 clinical trial for two indications, including first-line CMML, and also refractory relapsed classical Hodgkin lymphoma. So for this molecule, we are conducting IIT study for cardiovascular disease. So another one is IMM0306, which is a CD47 CD20 bispecific. We are finished enrollment for phase 2 clinical trial for refractory relapsed follicular lymphoma, and we just filed the application for phase 3 clinical trial. And at the same time, we're also conducting phase 1B clinical trial for two autoimmune diseases, including SLE and also NMOSD, which I'm going to bring the data later. So this is the first one, which is CD47 targeted molecule. So the earliest phase 3 readout will be come out in Q4 of 2026. And also we're conducting IIT study for atherosclerosis. So this is the MOA of this molecule. Our molecule in one way can block so-called don't eat me signal, and also we have an active Fc because our molecule was engineered not to bind the RBC at all. So we can have this active Fc, which is actually required for full activation of macrophage. If you delete the Fc function, then you lost anti-tumor activity in vivo. Meaning it's not enough for just only blocking so-called don't eat me signal. Sorry. So yeah, just bypass this market share. So this is the data we already have and also presented in ASCO and ASH in 2024, which is our presentation. And you can see that the data actually was very encouraging. So based on this data, we are encouraged to move forward to phase 3 clinical trial. So this is for first-line high-risk MDS, and also the data is very encouraging. But for this indication right now, since we have a limited budget, we just didn't initiate this study for this indication. And this is the data for safety data from the phase 2 clinical trial. And this is for first-line CMML, which is, which is also phase 2 clinical data. So basically, the ORR is over 70%, and CR over 27%. And also, the safety data is also well managed and well tolerated. So this is another phase 2 study for relapsed refractory classical Hodgkin lymphoma. The data is also encouraging, with ORR over close to 70% and CR 24%. And this is the safety data, which is also very well tolerated, not like CD47 antibody, which is because of the high affinity, it can confront a very severe so-called antigen sink. So you have to increase the clinical dose. So if you increase the clinical dose, for example, by 20 or 30 mg/kg, then the patient cannot tolerate. That's why we probably most of you know that one antibody from Gilead, which is magrolimab, called magrolimab, they all failed for the phase 3 clinical trial because of the toxicity. So for this indication right now, we are currently conducting phase 3 clinical trial, and which will be finished by end of next year. So this is the clinical development plan for phase 3 trials. And also for this molecule, we are conducting IIT study for atherosclerosis. So this data is from literature published in Nature in August 2016 by Professor Wenzhi's lab. And you can see that CD47 is overexpressed in the red area of the plaque, and in comparison to the normal tissue. And also for the plaque, if you use anti-CD47 antibody, so you can activate macrophage to eat up, to phagocytose the plaque, so you can get rid of the plaque. So based on this, Dr. Wenzhi, they set up a new company called Biotech Bio last year, which they raised $142 million for that molecule called like BRB02. However, if you compare the molecule, there is also subpart of Fc fusion protein. However, because of their molecule bind the human RBC and mouse RBC, they cannot use active Fc. So that's why they just delete the Fc effector function by engineering. So recently we finished the animal study using this animal model. And you can see that on the right-hand side, on top, with 2 doses in comparison with the vehicle and also BRB02, you can clearly see that our molecule, together with the BRB2, can all get rid of the plaques. Although, BRB2 is less effective than our molecule. So that is understandable because BRB2 only can block so-called don't immune signal, because without inactive Fc, they cannot fully activate the macrophage. So for this study, actually, we have enrolled 5 patients, and we anticipate the readout sometime in June next year. For this study, which is a RCT study, and including 32 patients. So I stop here for this molecule. The next one is CD47, CD20. So we finished phase 2 enrollment for follicular lymphoma, and also right now we are conducting phase 1b study for SLE. And in this molecule, we add the CD47 binding domain to the N-terminal of the heavy chain of CD20 antibody. Because that CD47 binding domain has been engineered not to bind RBC, so this molecule, just like the first molecule, also cannot bind RBC. And this is the data for follicular lymphoma. We just accumulate the data. The ORR is over 88%, and the CR is almost 53%. So for this indication, we already filed phase 3 clinical trial application in China. So this is the toxicity safety data for phase 2 study, and also we are very confident this molecule is very well tolerated and well manageable. So for SLE, since we already observed during the phase 1 clinical study, our molecule, even at a very relatively lower dose, as low as 0.5 micro per kg, this molecule can completely deplete peripheral blood B cell. So this is the expression of these two molecules, CD47 and CD20, in different lineage of the B cell. So you can see that for CD20, CD20 is absent in the plasmablast, which is a very critical B cell lineage for autoimmune disease, in particular for SLE. Because of the absence in this cell lineage, that's why rituximab actually failed earlier, failed phase 3 clinical trial for lupus nephritis. This molecule based on CD47, which is expressed in all of the cell lineage. So our molecule can cover all of the lineage and to get rid of the cells from the tissue and also from the blood. So in China, we already have IND for 3 indications approved. We are planning to file IND in US and also in China for several other indications. Yeah, bypass this market size. This is our clinical development plan, which is ongoing, and for SLE. So we choose 3 dose cohort, 0.8, 1.2, and 1.6 micro per kg. And right now we are at the last dose, 1.6 micro per kg. And we already finished the 5 patient enrollment. So the last one is waiting. Sorry. So the data is very encouraging. So this is a summary of the efficacy for 2 dose level. For the third dose cohort, we are still waiting. So you can see that clearly there's a dose-dependent efficacy response. And also for different symptoms, including arthritis, rash, and vasculitis, and alopecia. So this is the B cell depletion data. So you can see that from the beginning of infusion and end of the infusion, the cell can be quickly depleted from the peripheral blood. And also, you can see that some of the patients got rebound for B cell. For this B cell, actually, when we analyze in detail, it is a naive B cell, meaning the patient just can get recovery for the immune function, meaning the disease B cell has been deleted. So this is initial data from our study, and also in comparison with several other 3 molecules, including the belimumab from GSK, and also another one, telitacicept from Chinese company, and also from Roche for the CD3, CD20 TCE. And our efficacy data actually is the best, although the patient number is relatively low. So based on this data, I think our molecule has great potential because we call this molecule is MC, is a macrophage cell engager, which does not release IL-6, meaning this molecule will not induce cytokine release syndrome. This is the BD summary for several molecules in this area, especially the first one, which is also MCE. And Sanofi acquired this molecule. For the upfront payment is $600 million, and the milestone payment was $1.3 billion. And the last molecule, which is active receptor 2A Fc fusion protein, is right now currently under phase 1 clinical trial in China, targeting PAH. So this molecule, in comparison with sotatercept from Merck, our molecule has several advantages. For example, our molecule is very homogeneous and which is stronger, 5 times stronger blocking activity to the ligand and the receptor interaction. So we anticipate better clinical response, including safety and also efficacy. I think I'll stop here. So this is the animal study data. I won't go through that. Yeah. So this is another asset at early stage, which we will plan to file IND early next year. Okay, thank you. I just stop here. Okay. Yeah.